Hey, welcome to this last module of the course. My name is Avi, and we'll be talking about memory. And this is a very fundamental aspect of the nervous system, and in some sense, it's the part that gives us our own identity. Because say if you woke up tomorrow and you were blind, you'd be very impaired, but you wouldn't lose your identity. But if you lost your memories, then you would be completely lost. You wouldn't know where you are, where to go, what to do. And we know surprisingly more than you would think about how memory works. So this is what we'll be talking about in this course. And first, we'll talk about some history of memory research. So how is it that we got to the current intellectual point we're in now? So some of the earliest memory work was done by René Descartes, who was a famous philosopher and mathematician that you might have heard about. And he came up with this idea here, which is that he got this view that there was some experience that happened and that somehow was stored in mental tissue. So he imagined this mechanism here, which is think about a cloth that's spread out and then someone punctures it with some needles. And that's the kind of experience the cloth is going through. And then if you remove the needles, there are some holes left over in the cloth, and from that you can infer the relative positions of the needles that made the holes. So it's like this previous experience of being punctured by the needles can be read out by this hole pattern. So he imagined that something like that might happen in the brain, which is that in the brain he thought there were these fluids called animal spirits flowing through the brain, and then you had some experiences, and then those fluids made little holes inside different parts of the brain, and then that's how we would store these memories that already happened. And that evidently didn't turn out to be true, but there were some important conceptual advances in this theory that really were influential. And the parts that are correct in this theory are so basic and well accepted now that it's very difficult to see what was correct about this theory. So does anyone have a guess of what is correct in this theory? Yep.
Is it possible simply that the brain changes as we move things?
Yeah, so that's one thing that he got correct, which is that the brain physically changes in some way to kind of encode some experience. Yeah.
Regarding synaptic plasticity of the wires of the brain, it's also the dendrites is also the synaptic plasticity increases as we actually have memory traces that are happening throughout our daily life.
Yeah, that is true that we do have synaptic plasticity, but at this moment there was no synapse, no plasticity, so they didn't know when it. So this theory is not even wrong in that regard because they didn't even consider that to be a thing. But the most basic thing that this theory got correct is that he thought that the memories were somehow stored in the brain, and that was some very big conceptual advance that really turned out to be true. And because of that, he had this diagram that you have this brain, and then these animal spirits are going through it, making holes, and then that's what your experiences were stored in. And after that, another conceptual important advance happened with this distinguished looking person here called Richard Simon. And he had a very kind of colorful life. So first he grew this impressive mustache, then using this mustache he seduced the wife of his co-worker at the university, and then but how could he resist? Look at this. And then he got fired from the university, so he was very sad. And then after he was sad, he began writing about memory. So he published an important book called The Mem, and that had two important insights, which we'll see in the next slide. And then after that, he finished writing his book of memories, and then he was from Germany, and Germany lost World War I, and then he got very sad with that, and then he died in the bed over the German flag in a very sad way, and little did he know that Germany would lose another world war soon after. Okay, and he shot himself because of this defeat. But what did he do after he seduced the professor's wife and before he shot himself on the German flag. That's why he is remembered today, which is that he wrote this book in which he came up with these two important concepts. The first is the concept of the engram, which is a idea that kind of follows from that Descartes experiment, that there is some kind of physical process that happens in the brain that changes the neural substrate in the brain, and then that's how the memories are somehow stored. So he came up with this name of the engram, which is the physical memory trace. And then he also came up with this concept of ekphory, which now in more modern terminology, we use the term retrieval, which is you store the memory somehow, and then you have to retrieve this information to use it later. So he came up with these two important concepts, and then he shot himself over the sadness of the war defeat. Okay, so then the next important conceptual advance was with this gentleman here called Santiago Ramon y Cajal, who also had a very colorful life. And he was the founder of the neuron doctrine in neuroscience. The neuron is the fundamental computational unit. And he didn't start out as being a model student, so he was very mischievous and sort of a Spanish hooligan of sorts, and disrespected his elders, got kicked out of school, and then in his free time he built a cannon and blew up the gate of his hometown. And after his cannon blowing up days were over, he turned his attention to neuroscience, as all young cannon enthusiasts do. And then when he was in neuroscience, he came up with the idea that the synapses are where the communication happens between neurons, and then the changes in the strengths of synapses might be where experiences are stored, which is very, very far-sighted for that time, because you couldn't really kind of measure or see how the synapse was functioning. So he deduced all of this just based on anatomical tracing and looking at things in the microscope and so on. Okay, so then after that we go to the 30s, where we have this person called Carl Lashley. And Carl Lashley started the search for the engram. And then we'll also talk about these other experiments by James McConnell using planarian worms and Agranoff using fish. So here we start with Carl Lashley. And he was a person, I think, at Yale in the 30s and 40s that was one of the first people to study systematically which part of the brain might contain and store these memories that everyone was talking about. And he used very kind of systematic, rigorous experiments to study this. And at that time, in 1929, he used this technique of brain lesioning from stereotactic measurements, which was then a very new technique of brain lesioning. And that was invented by Shefford Franz, who was the person that we honored here in this department by naming this building the Franz Hall. And he concluded things that turned out not to be correct, but it was an important point to show that it was possible to rigorously study memory in animals. So to do that, he used this method called stereotactic surgery. And here, what you do is you use the kind of fusing points of these plates in the skull to figure out where brain structures are. So as you might know, our skull is not just one bone, but it's rather the fusion of some plates. And then in our head, these plates are very hard, so they don't move. But in babies, the plates are kind of very soft, so their heads are squishy, which makes it easier for them to go through the birth canal. But in us, it's hard. And so in these locations where the brain plates fuse, so you can see there in the front, there's a point where 3 brain plates fuse. We can use that coordinate to determine a system to figure out where other brain regions are. So we have already mapped out the coordinate position of all the brain regions relative to this point where the brain plates fuse. So it's kind of like, oh, this structure in the hypothalamus is 7 centimeters to the left, 8 centimeters deep, and 2 centimeters to the back of this point. So using kind of this 3D coordinate system starting from this point of the fusion plates, we can figure out where all the brain regions are. And one example is shown here. We have a rat brain, and this particular nucleus is, like this many millimeters to the left, this many millimeters to the bottom, etc. So you can do this to very accurately lesion different brain regions because you know where they are. And this was the intellectual background on Carl Lashley was making these brain lesions. So it was already known that the neuron was the fundamental organizing unit of the nervous system from Cajal's work. And we also knew about synapses governing simple behaviors. And this was done by Sherrington, who was an English neurophysiologist who figured out all those spinal reflex pathways. So we knew about synapses controlling those behaviors. And we knew this potential hypothesis that changes in synapses might be storing memories from also Cajal's. But at that point, this was an idea. It wasn't really verified experimentally. And people had already started to make labs and study psychology and memory in rigorous ways. So we have Wundt, who was the first psychology lab in 1879. And then there was this fellow called Ebbinghaus who made the first behavioral studies of memories. So he studied on himself because no one else wanted to participate in this. So he basically memorized long lists of words, and then he took careful annotations of how much he has to memorize to remember something in the future, how much he forgets. And then he came up with these concepts of learning curves and forgetting curves and their relationship to training, and what are some mental strategies to remember better or worse. So he did the first kind of systematic behavioral memory studies. Then it was also known that the work of Thorndike with instrumental conditioning is also something that they knew, which is, I think something Professor Wasson already covered, but these instrumental behaviors are assays in which the animal has to perform a certain action to change some outcome in the environment. And lastly, Pavlov's work with classical conditioning was also already done. So this was the kind of scientific background at that time. And the most recent thing that was happening was the rise of behaviorism, in which psychologists like Watson and Skinner were concentrating just on behavioral outputs and ignoring anything that happened inside the brain because those were kind of not directly measurable. So behaviorism didn't really concern themselves with the engram, which is the physical trace of the memory, because that's something that's inside the brain and the mind that you can't access, so they weren't concerned with that. So this was what kind of was known in Carl Lashley's time, and he used that stereotactic lesioning technique to make lesions in rat brains. So the lesion area is shown in black, and sometimes the lesions were very big, so it's hard to understand what's happening with a very big lesion. But what you can appreciate here is that he was very careful and meticulous in kind of writing down where the lesion happened, what's the extent of the affected area, and what was the change in the behavior. And the behavior was studying these mazes here. So there's a start and finish point, and you see the mazes have different amounts of complexity, and the rat can learn to do this. But the issue is that because the lesions are sometimes big and encompassing many different cortical regions, the reason for doing a bad performance in the maze doesn't necessarily have to do with memory. Right? So for example, if the lesion went to the visual cortex, then the rat would be bad at doing the task because he's kind of blind, not because the visual cortex is important for learning and memory. And because of that kind of problem, he found this result, which is that the more cortex is destroyed, the more errors happen in the learning of these mazes. And also similarly, if after the rat learns and then you make the lesion, then also he found the same thing, that more cortex destruction creates more errors to relearn the task. So he came up with this graph here that shows that the amount of errors increases with the amount of cortex destroyed. But because the lesions are very big and not very well controlled, he came up with this kind of incorrect conclusion that memories are kind of uniformly stored across cortex, so there's no region that's particularly more important for storing memories. And because of that, he came up with these two principles, which are equipotentiality, that all parts of the brain have the same potential or equal potential to store memory, and that there was this mass action principle that lesioning more of the cortex just creates more memory deficits, because he thought there wasn't any region more important in memory than others. And this was in contrast with this other important movement that was going on at that time, which was phrenology, where they thought that different little cortical regions that are more or less robust in different people created different bumps in their skull, which determined their personality. And this is also an incorrect view, but there was this view that there was functional localization at that time with the phrenologists, and so this conclusion was against that. And then it suggested that this search of memory was a dead end, because these big lesions created these results that were difficult to interpret. Okay, so what are some other ideas that happened? Another idea that happened was that memories may not be stored as structural changes in specific parts of the brain, because Lashley's results suggested the whole brain is uniformly storing these memories. So another idea was that perhaps there's some molecular basis to memory, where experiences might be stored in changes in the genetic material. Because at that time, we already knew the structure of the DNA and knew that was a biological way to store information. And from that idea, we had these cool experiments by James McConnell, who studied memory transfer using the planarian worm, which is kind of a flat worm that's shown there and has two little funny cartoonish eyes. And that's the planarian worm. So what they found out is that you could teach the worm a certain task, which is this task here. The task is basically you pair a light that the worm detects with its funny eyes, and then when the light comes on, you give a shock, then the planarian is very sad because it's shocked, and then it runs away. And then it learns that when the light happens, I should run away because a shock will happen. So the worm can learn this. Then they found out this very strange thing, which is that they found out that if you got a worm that learned this task and then killed that worm and then fed that worm to a new worm, then that new worm required fewer trials to learn this task. So it was this idea of learning through cannibalism, and then they thought there's maybe some molecule inside the worm that's being transferred when the other worm eats it, and then it gains its knowledge. And this turned out to later be found out that there was some evidence that that was some RNA molecule that this learned worm was producing. And there's some studies even more recently here at UCLA showing that some other invertebrates might also use RNA as a form of storing memories. But this hasn't been shown in mammals. So don't try to eat your friend's brain to gain their memories. It's frowned upon. Okay, so then we have some other scientists that started studying memory in vertebrates like fish. And here the task is there are these two parts of this compartment, and then the fish goes to one part, and then a red light comes on, and the fish is shocked, then it runs away to the other compartment, and then it's here, and then a red light comes again, and he's shocked again, and then the fish learns that when the red light comes on, I should move away because otherwise I'll get shocked. So that's the kind of shock avoidance paradigm, pretty easy to learn. And the fish can learn this, and the fish can then remember to do this the next day. So the fish had some kind of long-term memory of this. Okay, so then they had this idea of let's try to interfere with the fish learning this task. And to do that, they injected puromycin, which is a protein synthesis inhibitor. So they injected this drug inside the brain of the fish. And here's what they found. So what they're plotting, the y-axis is the retention score. So 0 would be kind of the baseline normal amount of retention for a healthy fish. And then they train the fish to do the task, so the fish learn how to do it. And then they injected this protein synthesis inhibitor at different time points after the learning had happened. And then they tested the effect of that injection the next day. Okay, so they trained the fish, then they injected the puromycin 0 minutes, so right after he ended the training. And if you do that, the next day the fish is very bad at remembering the test. The retention score is bad. And in another fish, they trained the fish, then waited like 60 minutes, and then injected the protein synthesis inhibitor. And that fish is not as bad the next day. So there's something happening right after the learning has occurred that is inhibited by this protein synthesis inhibition drug. And then they also found that if you were to inject the protein synthesis inhibitor the first time the fish was learning, so prior to the initial training, then the fish did learn to avoid the shock during the initial session, but then it didn't remember how to do it the next day. So there's something here that requires protein synthesis in the beginning and not like after 90 minutes. So this created this very important biochemical insight. It's maybe the first biochemical insight into how memory works, which is that the consolidation of the long-term memory requires protein synthesis. And this turned out to be true not just in fish avoiding red lights to get shocked, but also in mammals learning all sorts of other different things. But it also showed that there are different mechanisms that are mediating short and long-term memory, right? Because if you inject the protein synthesis inhibitor just prior to the initial learning, and he was never shocked, he learns how to avoid the shock at the end of this session. But then doesn't remember on the next day. So it shows that the protein synthesis is required for the consolidation of the memory, for becoming a long-term memory, but it's not required for this initial short-term learning. And this also creates this important division between short-term memory and long-term memory, and showed that they require kind of different biochemical pathways. So any questions so far about this? Okay. Yeah, you can just feel free to raise your hand and ask at any moment. Yeah.
So to clarify, the same day they did the injection, right afterwards the fish had trouble remembering the shock and to avoid the shock, but within that same session, say 90 minutes later, it could remember to avoid the shock?
Yeah, so they did two experiments. The first is this fish never did any behavior, it's just here. Then you inject the protein synthesis inhibitor, then you go to the training, the first initial training, and then he learns, okay. But then that fish the next day can't remember what to do. So this initial acquisition doesn't require protein synthesis. Then the other experiment is, again, a fish with no experience, then no injection, then the fish learns the behavior. Then after the behavior is learned, they injected the protein synthesis inhibitor right after the learning happened. And then the next day he can't do it. But if you just learned and then you wait like 90 minutes and then do the injection after the learning, then the protein synthesis seems to have already have happened, and inhibiting protein synthesis later doesn't seem to have an effect. So the protein synthesis doesn't seem to be required for maintaining the memory, only for the initial consolidation. So from this very simple experiment, three very big insights came about: that there is a difference between short and long-term memory, that the long but not short-term memory consolidation requires protein synthesis, and it also established that there are these two sorts of memories that are different. Yeah.
This inhibitor, is it like a transient thing, or is it a long-term, it's just knocked out completely?
It's transient, yeah. It's not inhibiting for days. It's just for some minutes, because it's kind of getting washed out of the brain, metabolized, etc. Yeah, you had a question?
For the short term versus long term, how do they differentiate? I know this is a little bit asked before, but how do they test the two of those?
Oh yeah, here they differentiate the short-term and long-term, not super rigorously, but just based on the initial learning versus remembering tomorrow. And remembering tomorrow was retrieval, or ekphory, as invented the term by the mustache guy that committed suicide. Okay, so, and now we have other sorts of divisions for memory, and some of these have already been covered by Professor Nantya Sutana, so I'll just go through these more quickly. But as she already mentioned to you guys, there's at least two different types of memory that you can classify. One is non-declarative or procedural memory, which is knowledge of how to do things, such as how to ride a bicycle, how to play tennis, etc. So those are kind of motor actions. And then there are these declarative memories, which are memories of what? And declarative memories can be either of your own life experiences, like yesterday I had pizza for lunch, or it can be also facts that you know, such as pizza has cheese in it, and then that's not part of your experience, but it's kind of a fact that you know. And I think she might have used exactly the same slide, but you have this kind of broader taxonomy of memory within which there's long-term memory, and you can divide this from declarative and non-declarative. And the declarative part, which is the part we'll focus more, we can further subdivide them into episodic memory, which are remembering episodes of your life, and semantic memory, which is knowledge of facts of the world that are not tied to your particular experience. And within non-declarative memory, we have learning skills, such as playing tennis, priming, which is being influenced to use a word that you just heard, and conditioning, like Pavlovian conditioning. So then we go to the more modern studies of memory, which is also something that Professor Nantya started covering. And as she already told you guys, a lot of these discoveries happened with patient HM, who was a patient who had intractable seizures. And then to get rid of the seizure, the doctors removed some parts of his medial temporal lobe. So here you see there's the little holes in the medial temporal lobes. And then after that, he was cured from the epileptic seizures, but he developed some weird memory impairments, which were studied by these two very influential neuroscientists, Scoville and Brenda Milner. So Brenda Milner is kind of a foundational figure in the neuroscience of memory, so she's kind of the grandmother of all memory research. And here we have a picture of patient HM just standing a sunny day with no hippocampus, remembering nothing. And then he taught us a lot about how the hippocampus works. So the hippocampus has this name because it's sort of shaped like a seahorse, and hippocampus is the Greek word for seahorse. Okay, so the memory impairment that patient HM had were two. So first he had anterograde amnesia for declarative memories, which means that he couldn't acquire new declarative memories. So if you told him today that Paris is the capital of France, then he can repeat that now, but he won't remember it tomorrow. So he couldn't acquire new facts, and he couldn't acquire memories of new life experiences either. Like if you said, "Hi, I'm Avi," then he can follow that during a conversation, but then tomorrow he wouldn't know. So I have to introduce myself again. So he loses life experiences at all also. And then he also had temporally graded retrograde amnesia, which means he also lost those same types of declarative memories for periods that happened before the surgery, but that occurred in a way that's graded with time. So things that happened right before the surgery has no recollection at all. And things that happen 5 years before the surgery, he remembers a little. And then things that happened 30 years before the surgery, he remembers very well. So this suggested that super old memories are not stored in the hippocampus because he remembers those very well without having a hippocampus. And it also shows that the hippocampus is important for both storing somewhat recent memories and acquiring new memories that are declarative as well. And another thing that the patient had is that he had normal procedural learning. So he could learn new motor skills as well as anyone else. So he was able to learn new things like learn how to juggle or learn how to ride a bike or learn how to do this complicated motor action. So he could learn those as well as other people. So he didn't have any motor skill issues. And that created another important division, which is that procedural memories do not seem to involve the hippocampus, and thus different memories seem to have different neural substrates that are necessary for maintaining and creating them. And because he couldn't acquire new episodic memories, the doctors that were working with him had to reintroduce themselves every day. And there is a movie that's kind of based on a character like this. So there was this Adam Sandler movie called 50 First Dates, in which Adam Sandler has normal memory, but then Drew Barrymore is a character that has this patient HM types of symptoms. So she always thinks it's this particular day in her life, which happened before some accident, and then she can learn new motor things. But she doesn't remember what happened before the accident in a temporally graded manner, and she can't acquire new memories. So Adam Sandler sees Drew Barrymore, she's like, oh, she's so cute, I'll go and do my goofy Adam Sandler jokes to her. And then she's very charmed and likes him, and then the next day she doesn't remember him. So that's why the movie is called 50 First Dates, because they kind of had to reintroduce themselves multiple times. And then later I found out that's apparently based on some real story of some real lady who had this kind of problem, and she had a real relationship with the person that had normal memory, and the movie is based on that. Okay, so when Brenda Milner studied patient HM, they also made him try to learn new motor tasks such as mirror tracing, in which you have to trace this star while not looking at your hand, but looking at the reflection of your hand in a mirror. And then this turns out to be very difficult. But you can train yourself to do it, and so can patient HM. And patient HM was able to learn this new motor task as quickly as other people, showing that the hippocampus is not important for this kind of learning. And this established the first super important findings, the systems neuroscience study of memory, which is that the hippocampus is a central component of the circuitry underlying memory. Because remember, before this, we were at this dead end where Lashley had said, oh, the brain is the same, there's no part that's particularly important for memory, so everyone was very sad. And then here we have an example of a human patient that has a very specific damage to a particular region in the brain, and then he has a very particular impairment, which is impairments in declarative memory only. So it's a very specific lesion producing a very specific result, and then that ushered a whole new age of researchers pouring lots of attention into the hippocampus and how memories work in the hippocampus and so forth. So after patient HM, people started studying the hippocampus in animals. But it's very difficult to study declarative memories in animals because they're not very good at declaring stuff. But all the mammals have a hippocampus, and you can make them do tasks that require information that's declarative-like. And some of these early studies were done in primates using the delayed non-match to sample task. So the task is you have this monkey, and then you put an object in front of the monkey, like a key. Then the monkey lifts the key. And underneath the key, there's some food. So the monkey eats the food. And then the monkey has to remember the food was underneath the key. And then the monkey is faced with a key and a new object, which is blue circle. So now the rule is if the food was under object 1, now in this trial the food is under object 2, which is the non-key. So then the monkey, oh, I'm a smart monkey, so this food is under the other object, and he gets the reward there. So here the monkey doesn't have to declare anything, but he kind of has to remember in a declarative type manner where the food was. Like the key was the object 1. So this was a very early attempt to study declarative memory in animals. And then they found that if you lesion the hippocampus in the monkey, then the monkey is worse at this memory task. And then if you lesion the hippocampus plus these cortical regions that are interconnected strongly with the hippocampus, such as the entorhinal cortex, then the monkey really is very bad at doing this task. So it showed that the hippocampus and adjacent cortical regions that are well connected to the hippocampus are super important for these declarative tasks of sorts. And these are kind of the regions that were also destroyed in patient HM surgery. And then after that, people started studying the hippocampus in rodents. So here we have the hippocampus in the human brain and in the rodent brain. So in the rodent brain, proportionally, the hippocampus looks much bigger. But that's mostly because in the humans, the cortices are very large, so they seem to be very overblown. But the rodent also has a big hippocampus. And another cool thing to notice is that the rodent has a big olfactory bulb here, while the human has a very tiny olfactory bulb that's not even visible in this picture because different animals view the world differently. So rodents use a lot of smells, so they have a big olfactory bulb. Yes?
I have a question about, 2 slides ago, you said that animals do not have declarative memory.
No, I said they don't declare.
Oh, okay.
But no, they have, but you can evaluate declarative memory in them through these sorts of tasks.
Okay, so along with that, so for example, let's say you train a cat or a dog, they, how do you differentiate between them, demonstrating some kind of memory retrieval versus them, doing a permanent conditioned response?
Versus doing what?
A conditioned response.
Okay, because in this case, there's nothing to condition to exactly, because you're not associating like a tone with a shock or or a smell with a food. So there's no association here, but rather they have to remember this abstract rule. And the rule is not the food is under the key, because it could be a different object in another trial. So the rule is the food in the next trial is in the new, under the new object.
Okay, I see. Okay, so this task requires them to, they need to learn a rule.
Yes.
Rather than just,
Yes. The rule is not the food is first under the key. The rule is the food will now be under the novel object, which could, and another trial, the key could be the novel object. Yeah, so this term declarative memory has to do kind of with how most humans operate, which is that, we are able to declare these memories. But if you think about it a little deeper, that's not really what the declarative memories are, because say if you're unable to speak for some reason, you don't not have declarative memories. It's just that you can't declare them, but you still have your life experiences and are able to learn new things. So it's somewhat of a misnomer, but makes it easy to understand what they are. Yes. And other studies that are earlier about the rodent hippocampus were done by Cajal after he did all the cannon gate blowing up and etc. And then he did very careful histological examinations of all sorts of brain tissue, including the hippocampus. And he was the first to notice that the hippocampus is very well organized. So you see there are all these layers and axons going from one layer to the other, suggesting that the hippocampus has some functional differentiation. So every time in biology you see that, this part of the tissue is a little different from that other part of the tissue, that almost always indicates that those parts have different functions. And by looking at the hippocampus, he figured out also the direction of flow of information within the hippocampus by looking at the direction that the axons are connecting to. So he'll do these little arrows showing that, the axons from this cell go to here, the axons from that cell go to that next cell. So from this was able to pretty accurately infer what's the directionality of flow of information through this hippocampus. And some very important research on the hippocampus happened right here at UCLA, where in Professor Michael Fancelow's lab, they discovered the role of the hippocampus in fear conditioning. So the fear conditioning experiment is basically you get this rat, Then you put them in a box. Then the rat's like, oh, it's a box. I'll look around. I'll rear heel a little. I'll smell this corner. So the rat's not afraid of the box because it's just a box. And then a tone happens, like beep, and the rat gets shocked. Then another tone happens, beep, the rat gets shocked. So the rat then learns to associate two things. The first thing is, well, I've never been in this box, and I was there, I got shocked, which was very unpleasant. So this box is some predictor of the shock. And the other thing is, when I hear this tone, I got shocked. So the tone is a very good predictor of the shock. So the rat learns both this contextual fear, that when I'm in this box context, it's maybe dangerous, and he learns that this auditory tone also predicts the shock. Okay. And then the rat remembers this for like many, many months, essentially for the whole lifetime of the rat. So after this experience, you put the rat back in this box even like a month later, the rat's like, oh, it's this box. Something horrible will happen at any moment. The rat freezes in fear. And so the rat exhibits this freezing in the box. And then if you put the rat in another different box that he never seen before, he's like, oh, it's a box, it's nice. And then you play the tone in the new box, then the rat's like, oh my God, it's the tone again. So the rat freezes when he hears the tone. Okay, so then at UCLA here in the late 90s, they decided to test if the hippocampus had some role in these memories. So they found out that lesion in the hippocampus impaired contextual freezing, which was the association of this box is dangerous. So the rat freezes less if you lesion the hippocampus in the box that was scary. But it's also very interesting, the rat does not freeze less to the auditory tone. That very same rat, even. And this showed like a further division in the taxonomy of memory, which is that kind of contextual associations seem to require the hippocampus, while cued association seems to not require the hippocampus. So it's like we have this one word in English, memory, but that doesn't seem to really correspond to some neurobiological process. Rather, there are all these different sorts of memories that require different biochemical pathways in different parts of the brain. But this, along with some other data, showed that the hippocampus is very important for memories of contexts. And it also, going back to the patient HM's impairments, we knew that patient HM didn't have hippocampus, and he had early memories pretty accurately retrieved, like when he was a kid, but his more recent memories right before the surgery were more lost. And then that was observed with some other people that had amnesia. So this study here was done in the late 80s, in 1989. So 1980 is the recent time at that moment. So they got these people that had amnesia and people that don't have amnesia in 1989 and asked them about some public events. So they asked them, oh, do you remember when President Reagan was elected? Then they're like, what, the actor? He got president? I don't remember that. So they didn't remember things from the 80s. Then you ask them, do you remember the Vietnam War happened in the 70s? I remember there was a war of some sort. Then if you ask, do you remember Elvis Presley got famous in the 50s? Like, oh, yes, I remember. I like that Blue Suede Shoes song. So they remember things from the distant past, but not recent past. And the control people remember everything. And then that was also shown to be the case with contextual fear conditioning in rats. So here, in this experiment, that they showed that if you lesion the hippocampus, they don't learn this contextual fear conditioning. But this has an interesting kind of time progression, which is the following. So here that is this experiment where they conditioned the rat to this box. So the rat was placed in this box, then shocked in this box, and then the rat got scared of this box. Okay. And then they lesioned the hippocampus at different delays after the training had happened. So first they always trained the rat with no hippocampal lesion. So the rat learns this with a functioning hippocampus. And then after learning it, they lesion the hippocampus first, right after the learning. And then the hippocampal lesion rat doesn't freeze to the context the next day. Or they waited like 7 days after the training and then lesioned the hippocampus. And then the rat remembered somewhat that the place was dangerous, so froze a little more. But if you do the hippocampal lesion 28 days after the training, it seems like that didn't matter. Like if you lesion the hippocampus a long time after the learning, then the rat still remembers that this contact was dangerous and freezes there just like the normal rat. So this shows that the hippocampus, both in the rodent and in humans, are more important for acquiring and storing these more recent memories, while these older memories seem to not require the hippocampus. So the idea is that on day 28, these memories are somehow being stored somewhere else. So destroying the hippocampus at that moment didn't impair the contextual fear conditioning. And now we know this somewhere else is the cortex. So memories are being kind of gradually transferred from hippocampus to cortex. So we shouldn't think about this as the memory just sitting in the hippocampus and then it's day 28 then moves to cortex, but it's gradually being transferred to cortex, and then when it's like day 30, it has fully left the hippocampus and there's no memory in the hippocampus. And for people, that time frame is not 28 days, but it's more like 30 years. Yes?
So for this task, the kind of like dissociation is strictly like very negative. But have there been studies that have like showed this effect in more like positive, like reinforcement?
Yes.
Task? Oh, so it's like the same.
Yes, things that it's not any sort of task, but so they either have to be tasks that are kind of declarative type tasks or tasks that are context association dependent, right? Because the cue association ones don't seem to require the hippocampus at any time point. So yes, but it's valid for positive, happy memories as well. And then we can also see that from even patient HM symptoms, because it's not that he couldn't acquire new bad memories. He couldn't acquire new happy memories either. But this raises an important point, which is why it is that they did this bad memory test. And that's because both the rat and we as humans are much more prone to learn bad, dangerous things than good things. Because for survival, it's more important to be conservative and really know what's the danger, and it's like less important to know where to get free pie. And you can also see that in lots of people's tragic experiences, right? So there are people that, in 1969, this very bad thing happened, and now I'm depressed until now. So, but there's no opposite. There's no person that in the 60s, this wonderful thing happened. Now I'm happy because of that, like 50 years later. So there's none of that. And that's true of the rat, too. Like the rat, if you teach it contextual fear conditioning with a sufficiently strong shock, the rat will remember that like 2 years later, which is all the lifespan of the rat. But if you teach the rat that this tone predicts like ice cream, the rat won't remember that a year later. Sorry, you had a question here?
Yeah, I was wondering, I don't know if this is significant, but why for the sham group, their memory, or they were doing worse than the ones with the lesion? I was saying, like, if you have that lesion, you'll actually have better long-term memory.
But the sham doesn't have lesion.
No, I know. So I'm saying, like, how come they did worse after 28 days?
You mean the sham did worse after 20 days? So you're saying just looking at the rat curve, why it's worse at 28? Sorry, you're saying why the rat curve is different
I was
at...
just wondering for the sham,
Mhm.
when it, so like over time, when there's a greater amount of delay, like more days, the percent freezing went down, right?
Yeah, it goes down a little because it's a less recent memory. So they remembered that forever, but I mean, the memory is kind of more jarring and influential more recently.
But for the lesions, we're seeing opposite.
No, for lesion, like, we shouldn't think of them as the same rat, because it's like different rats for lesion 7, 14, 28, 28 days later. So on the rat lesion at 28 days after the training, that lesion just has less effect. So the lesion rat at 28 days is more sham rat-like. Yeah.
Why would it go up? Like, why is there more freezing?
Why does what go up?
For the lesion rats, why is there like more, there's an increase in
There's an increase in freezing because it's kind of the association that this context is dangerous. This memory is being stored in the hippocampus. And then that memory is completely stored in the hippocampus in the early days. So if you lesion the hippocampus, then there's just no memory. So then it's like, oh, it's a new box. Nothing ever happened to me here. So I'll just not freeze. And then by 28 days, a lot of that memory has been transferred to cortex. So there's this kind of other copy of the memory. So even if you destroy the hippocampus, then the rat's like, what is this box? And then even with destruction of hippocampus, he still remembers because the memory is not stored in the hippocampus. So, yeah.
So to be clear, the black line for the hippocampus lesion, like each of those points represent different animals that got the
Yes,
lesion.
yes,
So
yes,
it's-
yes.
So yeah, it's not the same rat.
Yes,
It's
yes.
better.
Yes, it's different animals. Yeah, it's not the same rat getting better. So that study kind of lesioned the whole hippocampus, so it was difficult to figure out what the different subregions of the hippocampus do. But as we saw from Cahal's early anatomy studies, the hippocampus does seem to have different subparts, so people naturally paid attention to probably these subregions have different functions, so let's see what they do. And you can see it's organized in some nice cell layers, and the main regions of the hippocampus that were studied were the dentate gyrus and area CA1 and area CA3, which are located there. So if you kind of, one way to think about it is that the hippocampus is two interlocking Cs. And then that two interlocking Cs are curved around each other like a banana. And that's the shape of it in 3Ds. Two pieces of tissue like this, and then you wrap it around like in a banana shape. And the connectivity of this region has been very well studied. So we know that the flow of information is sort of the same way that Cahal has inferred it was, which is that the main input of the hippocampus is this middle entorhinal cortex, which then projects to the dentate gyrus. The dentate gyrus projects to CA3, CA3 to CA1, CA1 to subiculum, and subiculum back to the entorhinal cortex. And this is what it looks like in a more simplified manner. And all of these projections are excitatory. So you have this excitatory cells in entorhinal cortex projecting to excitatory cells in dentate gyrus, which project to excitatory cells in CA3, CA1. So all of these components are excitatory cells that project to each other in a loop. And given that this is the connectivity, does that give anyone any idea of why is it that the hippocampus was removed as a focus of a seizure initiation point in patient HM? Like, is there anything about this anatomy that would make you think it could generate seizures? Yes?
Because it feeds back into itself and it's all excitatory.
Yeah, it's exactly that. So it loops onto itself, it's all excitatory. So if the inhibitory local cells are not working super well, you could easily get this excitation that excites itself recurrently and becomes kind of a looped positive feedback that grows and grows and grows and creates a seizure, and it can be very impairing. And that's why the hippocampus is one of the main loci in the brain that generates seizures, because you have this excitatory loop happening. Okay, and because this is such a well-studied region, each of the projections between the hippocampal subregions have their own little names, such as the perforin path, mossy fibers, and Schaefer collaterals. So because the hippocampus was already known at that moment to be important for acquiring new memories, people naturally wanted to study the neural connectivity in this region to see. I mean, we know that if you lesion it in this patient HM, he doesn't acquire new memories, but what's happening mechanistically, right? So you can't really understand that from lesions. So they started to study the plasticity of the hippocampus in a brain slice. And that seems like a very esoteric idea, because at least the patient HM is a real entity, an organism that can learn things, but here they're studying a brain slice. So they get a piece of the brain, so there's no animal anymore, and keep that piece of the brain with the hippocampus alive in a petri dish through kind of miracles of pharmacology for a few hours, and then they study the circuitry of that brain's tissue while it's still alive for a few hours. And there they found that you can kind of artificially increase or decrease the synaptic strength between these little hippocampal subregions using artificial electrical stimulations. And then you can induce both long-term potentiation or strengthening of connections, as well as long-term depression or kind of weakening of connections inside this little hippocampal slice. And that sounds like a very strange thing to do to study memory, but it's kind of very cool, because if you think about it, what's happening when you acquire a memory is you are there without this memory, then some experience happens, and then after some experience, your behavior changes because you learn some new thing. And they were able to do this in a little hippocampal slice without any animal attached to it. So they're able to electrically induce some experience in the slice, and then the slice changes its behavior after the electrical experience happens. And that's what we'll see in the next slides. And one person that had kind of theorized that this could happen was this very famous Canadian psychologist called Donald Hebb. And he predicted in 1949 a theoretical framework to understand these sorts of potential plasticity changes in the brain. So basically, his idea was that if one cell activates its output through an axon to another cell, and then that output has some effect on the output of the second cell, then the second cell would increase the influence synaptically of that first cell. So basically, that's shown here, which is, let's say we have this cell in the middle, which is a round cell, and the round cell has some kind of very weak synapse with the black cell. So when the black cell fires a little action potential, it's a very weak synapse, so then the round cell doesn't fire an action potential because of that, because the excitation is not strong enough. Now let's say some learning-related experience happens, where both cell A and B, which are the inputs to the round cell in the bottom, are active simultaneously. Now you have two excitatory little things happening on the middle cell, so the middle cell then is activated enough to fire an action potential. So if you do this a few times, then from the point of view of the middle cell, you see that, oh, whenever that cell fired, I also fired right after. So I should pay more attention to the input of that cell. So it strengthens the input of cells that are influential in its own behavior. And then after this learning experience, just one action potential just from the cell on the right is sufficient to activate the middle cell. So the idea here was that kind of cells that fire together, wire together, which is one of the most famous sayings in systems neuroscience. And here you can see this in a less abstract form if you think about the behaviors we've been talking about. So for example, in the rat tone conditioning experiment, what happens is, first the rat goes to the box, then he hears a tone, then the rat doesn't care because it's a tone. So the tone doesn't provoke some behavior in the rat. But then if the tone is paired with the shock, so it's like tone and shock, then he learns that the tone predicts a shock. So after learning that, then the rat changes his behavior to the tone, which previously he didn't. And that is sort of something that is explained by this kind of diagram. It's kind of before the learning. If you imagine the B cell is the cell that conveys the tone is a very weak input that doesn't cause this, let's say, freezing, creating cell to fire. Then you have some learning where you have some shock input and tone input happening at the same time, which produces a bigger excitation. And then after that, this synapse with the cell that conveys the auditory tone could become strengthened. And now the auditory tone by itself is able to activate the cell that maybe creates freezing. So this provides some kind of simple diagram that can explain associative learning in general. But at that point, yeah.
I just want to ask, why wouldn't you strengthen cell A's synapse as well, since that one leads to firing as well?
It would. For example, the tone example, the cell A is the shock conveying cell, but that's already kind of a very strong input, which is already sufficient to make the cell fire. But you could imagine that they are both two weak inputs. And in that case, if you simultaneously activate the both weak inputs, both of them would get strengthened. But if one of them is already super strong because it conveys something very important like a shock, then that one is already kind of strengthened previously. Yeah.
So this would explain like the long-term potentiation, or what about like long-term depression?
Yeah, we'll get to that in the next slides. Okay, so any questions? So, yeah.
Yeah, do we know if this is functioning based off of like a decreased saturation threshold of the postsynaptic cell, or is it like increased firing of the presynaptic cell?
So we know that there are both pre and post-synaptic mechanisms that happen in LTP, and then we'll cover that in the next slides. But yeah, you had a question? Okay. Okay. Any other questions about this? No. Okay. So, to study this neurophysiology, they made again these slices, and then we got to these questions that you guys asked, which are what happens in long-term potentiation and depression. So the experiment is you make a little tissue of the hippocampus that's placed in a petri dish, and it's kept alive with nutrients and oxygen and stuff. So what they do is they put a stimulating electrode in the Schaefer collateral. The Schaefer collateral is the name of the pathway of axon bundles from CA3 to CA1. Then they stimulate those axon bundles, and then you record the response in CA1. So CA3 is the input, CA1 is the postsynaptic cell. And if you electrically stimulate those axon bundles of the Schaefer collateral, you get a little kind of response in CA1, so nothing super exciting. And here, for like 15 repetitions, they're doing that. They're kind of doing a little stimulation on the axon bundle, then they're recording a little excitation in CA1. And if you do that for a bunch of time, you see that the response size is very conserved across trials. So nothing super exciting happening. And then they do something different, which is here at this moment, which is moment 0, they do what's called a high-frequency stimulation, which is they very, very strongly excite this axon bundle. So now instead of doing a little stimulation, they do 100 hertz of electrical stimulation, which is like super strong, and then they revert back to doing their normal little stimulation. So now something very astounding happens, which is that the slice went through this experience, which is high-frequency stimulation at 100 hertz, and now after it goes through this big dramatic experience of very strong stimulation, it responds differently to the little electrical impulse you were doing before. So after the high-frequency stimulation, the same electrical impulse that created a small response, this pink response, now that same stimulus creates this bigger response, which is this bigger blue response. So that happens, and that is kind of maintained for like several hours, which is the whole lifetime of the slice in the petri dish. And this was very exciting for people in systems neuroscience, because now it seems like you could study some kind of synaptic plasticity in a slice without any animal and behavior and other kind of complications of having a real living animal. But you might ask yourself, why do you want to study something in a slice instead of in a real animal that can actually behave and learn things, and this is not a thing that can behave at all? So you have lots of advantages when studying the slice. So one advantage is that because the slice is in the petri dish, you have complete biochemical control over its environment. So you can put more of this ion or less of that ion or some drug in the bath, or you can block a particular ion channel or agonize or antagonize another channel. So you can do lots of things to the slice that are difficult to do with the animal. But you do lose the kind of real behavior per se. Okay. And this was first found in the rabbit hippocampus, but later it was found out in all sorts of animals in all sorts of different brain regions. It was found that lots of different brain circuits, if you do this high frequency stimulation, that serves to strengthen the synapse between two different random brain regions, and now that synapse are strong for several hours. So it's able to maintain this kind of memory in the slice for essentially the lifetime of the slice. And someone asked about the long-term depression, and that too was found in the slice, which is that you can do those little test pulses, and then you do the opposite manipulation, which is low frequency stimulation at 1 hertz, and that seems to decrease the response size in a pretty stable manner, also for the entire lifetime of the slice for a few hours. So using, yeah.
What was the difference between the blue graph and the pink one?
Which what?
What was the difference between the blue graph and the pink graph again?
In between these two or between these two?
No, like in the waves.
Here?
On the waves thing.
Oh, here?
Yeah, like between the blue and the pink.
Oh, here the difference is that before the long-term potentiation, the response size that you saw was pink and small, and after the long-term potentiation with this high-frequency stimulation, this response became bigger, like this blue response.
Technically, like at the baseline of the stimulus itself, like before the stimulus happened for the pink one, and then like the stimulus happened, and then it gives the
Yeah, at baseline the response size was pink, and after the learning the response size was blue, which is bigger for long-term potentiation. And then the same thing is true here, where the baseline response is shown in pink, but then here after the kind of plasticity experience, the response becomes smaller. So the blue is always the post-experience response. But with high-frequency stimulation, the post-stimulus response becomes bigger, and then with the low-frequency stimulation, it becomes smaller.
So in the long-term depression, the blue one, which is the low frequency stimulation, is actually not as excitable as the baseline, because like the show of the graph is like the blue.
Yes, here it becomes smaller than baseline. So it's possible to also weaken the synaptic connection between CA3 and CA1 with low frequency stimulation, and you can also strengthen it with high frequency stimulation.
Additional question: why would this like record within the subscapula collateral?
Sorry, why would what?
Why would this simulation or experiment recorded within the scapula collateral instead of like different like sort of pathways within the hippocampus itself?
Why this was done in the hippocampus?
At the scapula collaterals instead of
Oh, why in the Schaefer collaterals? It was done in many other parts too. It was also done in the. That's just the pathway that has been more studied, but it has been shown to happen also in this perforin path and in the mossy fiber and the Schaefer collateral. So, and also many brain regions outside the hippocampus. The same LTD, LTP effect was observed. So the cool thing about this is that the first time they found this, it was in a hippocampus of a rabbit between two particular subregions of the hippocampus. So not super important in principle, but why it became important is that the same sorts of changes were found in other species and also all sorts of other brain regions that are not the hippocampus. And then it was later also found out that biochemical pathways that are required for these long-term potentiations in these slice experiments were also required for a real living animal to learn real experiences. So it provided some kind of in vitro way of studying synaptic plasticity. Yes?
So it's the same number of synapses that are being observed, but the synapse strength is what's changing.
Yes, exactly. Yeah. So this is not destroying the synapses, but it's making them weaker and stronger. So this is also something that was a big insight in these systems neuroscience works, which is that these synapses are not stable, but they kind of, in many brain regions, they can become stronger and weaker depending on the chemical environment and what the animal learned or didn't learn. But they change in a particular way. So all of this neuroplasticity tends to happen only in regions that already have some connection. Okay, so for example, in this case, they're studying the connection from CA3 to CA1. So projections that already exist from CA3 to CA1 can become a little stronger, a little weaker. But you won't learn something and cause some new axon from visual cortex to go into the hippocampus. So long-range connections are generally not being changed by plasticity. So you don't get new connections between faraway brain regions that didn't exist. But connections that already do exist, like this region is connected to that other region, those connections are very pruned and enhanced and a little micro-tuned and etc. Okay, so as we were mentioning, this was discovered in the rabbit, this long-term potentiation, by this very important paper by Bliss and Lomo, and then later this was found in all sorts of other circuits and all sorts of other animals and found to be important for other kinds of learning. So any other questions about the long-term potentiation? No? Right. So this was discovered in 1973 in that very important paper, first in the rabbit hippocampus. And it was the first time that they discovered some mechanism that showed a kind of biophysically detectable change that happened after an experience, right? Because until now, if you think about the whole trajectory of the intellectual development, they sort of thought that experiences would change something in the brain, but this is the first time that they actually found that here, there's a little synapse in this part of the brain that is strengthening and weakening from this electrical experience we are making the slice go through. So that was one big discovery. And the other was that before they were able to do some kind of synaptic strengthening with these electrical manipulations, but those manipulations produced only very short-term potentiation, like a few seconds or minutes. But here, if you see, the potentiation lasts for a few hours, so it's which essentially as long as a slice lasts. So this was the first time that some semi-permanent or very long time frame change happened in a circuit after some electrical manipulation. And as I just mentioned, this has been observed in all sorts of other regions in the mammalian brain, like the cortex, the thalamus, the spinal cord, like every random region you can imagine. And it also has been shown in other species as diverse as the octopus and the honeybee and the Aplysia, which is a type of like sea slug. And now, one important thing to remember is that this is not exactly memory. So LTP is studying synaptic plasticity, right, because they're strengthening and weakening synapses, but this is not the same as studying memory because this is not really happening in the real hippocampus in the same way that's happening in this slice. Because in the slice, what you're doing is you're electrically activating all of these axons from CA3 to CA1 in a very strong way with 100 hertz. And that doesn't really happen physiologically. So it's some weird lab procedure. But the reason why we think it's important is because a lot of the mechanisms that are involved in the synaptic plasticity that happens here is also observed in real learning in real animals through real experiences that are happening. So we don't have this kind of electrical activity happening in the brain where all the axon bundles are simultaneously activated, but we do have these strengthening and weakening of synapses that use these same mechanisms. And because of that, these mechanisms that are happening LTP are thought to be very important for learning and memory. Yes?
Are there areas of the brain that are more sensitive or susceptible to long-term potentiation? Or is it generally all just like the same?
Yes, that's a good question. So there are definitely areas of the brain that are more or less susceptible to this kind of synaptic plasticity. And because they're areas of the brain that are more related to learning and experiences and others are somewhat less, so it's because of that. And that also, the kind of biophysical synaptic properties are different in different brain regions. But this has been seen in many other sorts of learning in other brain regions. Yep. So okay, so we'll just get started here on the LTP, which is this experimental setup shown here, where you start with this. So imagine this is a brain slice in a petri dish, and then you have two different CA3 cells that are projecting onto that CA1 cell. Okay, so if you do LTP on the pathway from cell 1, after the high frequency stimulation, you get a bigger response on cell 1, which is the purple, before and after. But pathway 2, which is the other cell's axon that goes to the same CA1 cell, doesn't get strengthened. Okay, so this means that if you have one cell that receives input from, let's say, 2 cells, and you potentiate only the synapse from input 1, the synapse from input 2 would not get independently potentiated. So you have this pathway specificity of which synapse is being strengthened.
Would that occur because they're not both synapsing at the same time?
It depends what you mean. Well, the synapse is already there all the time.
No, sorry, you said for
Do you mean not firing at the same time?
yeah, like you said, one of them won't be like independently.
Yes. Yeah, the synapses are all there from pathway 1 and pathway 2. But if you strengthen pathway 1, pathway 2 is not automatically strengthened. And that is because of what you were alluding to, which is because the kind of downstream cell is trying to increase the influence of inputs that are predictive of its own output. So if cell 1's action potential is correlated with me firing action potential, then I'll be like, oh, I should pay more attention to cell 1 and strengthen that synapse. But in this case, cell 2 input was not active when that was happening, so the cell 2 influence is not increased. But if it were, and you were simultaneously stimulating 1 and 2, then both of them would get active, yes. Okay, so any other questions about this? Yeah.
How are they specifically measuring at the synapses? Like, how do they
You can stimulate like different sub-regions of CA3 which have different cells, and then you're just patch clamping from the same cell in CA1. So this is not something we really need to get into it, it's important to answer this question, which is that in this case, you don't necessarily need to be patch clamping a cell. You could just be recording the kind of bulk electrical response, not necessarily in one cell. So, but this also works if you record in one cell. But to do this thing, you have to record one specific cell. And then if you're recording this one specific cell, you can then electrically stimulate either kind of this sublocation of CA3 or this other sublocation of CA3 that have different cells. So if you stimulate this sublocation, then the axons from these cells will get strengthened, but not the synapses from here. Right? So it's kind of like you have the CA3 here, and then you have some area of CA3 there, some other area of CA3 here, and then you're patch clamping from this specific cell on CA1. And if you electrically stimulate this region, you kind of potentiate the synapses that originate from these cells. But you don't potentiate the synapses that go from that cell onto the same CA1 cell. So that's how it's done. Yeah. You had a question? Yeah, and then that's, yeah. This is mine and fell?
Did you read one? Because you've been pointing.
Oh, no, it's okay. I'll continue with my old-fashioned pointing ways. And then that's shown, the actual experiment shown here on the lower graph, where you see that if you induce LTP on pathway 1, you can get a long-lasting LTP there on pathway 1, but the pathway 2 doesn't seem to care about it because there's some anatomical specificity to this result. And the same is also true of LTD. So if you have two different inputs and then you induce long-term depression in one input, then the other input is not automatically depressed. The other input just continues as it was. So here, in this case, they depressed input 2, sorry, the blue input, but that doesn't cause a depression in the red input. And here again, the difference is that they used long-term depression by using 1 hertz low-frequency stimulation rather than 100 hertz high-frequency stimulation, which induces the potentiation. And here, depression is caused by slow stimulation at 1 hertz. Okay. Then another important development came when they studied this other sort of LTP called Hebbian LTP. Okay, so before what was happening is they weren't stimulating the green cell. They're only recording the electrical activity in the green cell, and they were stimulating the axon's inputs there. And then if you stimulate the axon input with this high frequency stimulation, you could get potentiation. But now they are pairing stimulation of the axon bundle with stimulation of the cell body of the green cell. Okay, so in this case, what they were doing in the previous experiments was they were only stimulating this, which is the input. And that produced this kind of LTP here. So they were not stimulating this cell. But you can also cause Hebbian plasticity, which is that you can stimulate this input and this postsynaptic cell simultaneously. And then you don't need to do this very strong 100 hertz thing. You can just do it at a lower rate. But that also strengthens this synapse now, because from the point of view of the cell, what it's trying to do is increase the synaptic strength of inputs that predict my generation of action potentials. So one way of doing that is if you stimulate the input super strongly, that will cause my action potentials to fire. So I'll be like, oh, this input is important because I got excited, so I'll strengthen this input. And that's what's happening in the high frequency stimulation case. But in this Hebbian case, what's happening is they stimulate the input not in some crazy 100 hertz, but some normal lower hertz frequency, but simultaneously they stimulate my own cell body. So now I see that, oh, this input fired action potential, now I fired an action potential, so this input is predictive of my behavior, so I will strengthen this synapse. And that's what this Hebbian LTP, because it kind of derived theoretically from that view Donald Hebb had of cells that fire together, wire together. Okay, so any questions about how this Hebbian LTP works, or what's the difference between this and the previous kind of LTP? Yeah. Sorry, could you speak louder? Oh, this is also a dish. Yeah. Yeah, so all of these things were done in dish, and somewhat more recently they were done in vivo in animals as well, but like 99.9% of these studies were just done in a petri dish. But it's like indirect effects of them can be seen in animals as well. So it's kind of, oh, doing LTP in the dish seems to require this particular little protein. And then you see that maybe this protein is important for learning in a real animal that has real behaviors. And then if you knock out that protein from the real animal, then the animal first doesn't show LTP in a slice, which is not super interesting, but more importantly, the animal doesn't learn real things as a functioning animal. So, yeah.
Can you circle the synapse that's strengthened when CA3 and CA1 are both stimulated?
Can you do what?
Can you circle the synapse that's strengthened? You said the synapse that's, the synapse that's strengthened. Can you circle it?
In this case?
Yeah.
Yeah, so,
Which one is strengthened?
oh wait, I do the, so here the thunder is not on this axon, the thunder is meant to be on the cell body.
So which synapse?
It would be this one. Yeah, so yeah, just to make it clear again, what's happening here is if you stimulate, let's say, cell 1 and cell 3 simultaneously, then the synapse from cell 1 to cell 3 gets strengthened, but not 2 to 3. And to do this, which is a Hebbian plasticity, you don't have to stimulate at some obscene 100 hertz, which doesn't really happen in real life. So you can stimulate at physiologically relevant amounts of frequencies, and then you still get the strengthening. Yeah, but the biggest explanatory factor is again this thing that you imagine that the cell just trying to increase the influence of the inputs that appear more already influential, right? So, and the influence is, does it make me fire an action potential? So if input 1 is active and then I'm also active, I'm like, oh, I have to strengthen input 1. So that's what it's trying to do in all of these cases. So there are these two important properties, which is the last thing we'll cover today, but you have this property of specificity that we mentioned already, but it's basically if you potentiate pathway 1, then this inactive pathway 2 is not potentiated, because pathway 2 is not predictive of when this output cell is being active. And there's this other phenomena that was seen, which was the associativity property. And here what they do is they stimulate pathway 1 strongly, like strongly enough to potentiate the cell. And now if you stimulate pathway 2 weakly, pathway 2 also gets strengthened. Because from the point of view of the cell, which is this third big cell here, when pathway 2 was active, I was also active, so I should strengthen pathway 2 as well. Even though most of the excitation came from pathway 1, what the cell is kind of computing is the coincidence in timing. Like right before I got active, pathway 2 was active. So pathway 2 must be important. So I'll strengthen pathway 2. And in this case, pathway 2 was inactive during this procedure. So when this pathway 1 was strongly activated, then the cell on the right was active as well because it's very strong excitatory input. But then pathway 2 wasn't active, so it's like, I won't care about pathway 2. So even if you weakly stimulate pathway 2 during a strong stimulation of something else, then this weak stimulation pathway will also be strengthened, which is this associativity procedure. Yeah.
I thought weak and strong had to happen at the same time, not a little bit.
No, they happen. Yeah, they happen at the same time.
Okay. Oh, okay. I thought you said that the weak like can happen a little bit.
Sorry.
Nothing.
Yeah. So yeah, they happen at the same time. So from point of view of the cell, it's like sorry. What I meant by a little bit after was the action potential of this output cell is let's say happened now, which is a little bit after these two things are active. So they won't be simultaneous, the input and the output of the cell. So, but the inputs are happening in close time proximity to its own output, then those inputs will get strengthened. Yeah.
Is it like correlated with like HFS? So for example, if we simulate the pathway 1 with the high frequency simulation and pathway 2 with LFS, the LFS on the pathway 2 is becoming strengthened by the pathway after that.
If you do high frequency stimulation on pathway 1 and low frequency on pathway 2,
The low frequency on the pathway 2.
Oh.
Is it going to be strengthened?
oh, that's a good question. I don't know an experiment that did that, but my guess is that maybe nothing would happen because you'd have competing processes, or it would get strengthened. My guess would get weakly strengthened. So yeah, his question was what would happen if on pathway 1 you did high frequency stimulation, pathway 2 low frequency stimulation at the same time. So it's a very good experimental question which I don't know the answer to, but my guess is that the pathway 2 would get weak, a little strengthened. And if you can just remind me again of this question next class when we're talking about the actual biochemical pathways, and then I'll explain why that's my guess that will get a little strengthened. But that's what we had for the first class, and then next class we'll continue with the actual biochemical mechanisms that are underlying these changes.